Hello viewers, good day to all of you. Today we are going to discuss on biomolecules. A biomolecule is any molecule that is produced by a living organism, including large macromolecules such as proteins, polysaccharides, lipids and nucleic acids, as well as small molecules such as primary metabolites, secondary metabolites and natural products. Biomolecules can be divided into two groups, that is inorganic and organic compounds. Inorganic compounds include water, mineral elements and mineral salts. Organic compounds are those which contain carbon and hydrogen along with nitrogen, oxygen, phosphorus and sulfur. Now we discuss carbohydrates. Carbohydrates are the most abundant biomolecules belonging to class of organic compounds found in living organisms on earth. Carbohydrates are poly, hydroxyl or OH groups and an aldehyde CHO or a ketone CO groups. Hence, these are defined as polyhydroxyaldehydes or ketones and their derivatives. Carbohydrates are linked with amino acid polymers, proteins forming glycoproteins and with lipids as glycolipids. These are a widely diverse group of compounds that are ubiquitous in nature. More than 70% of the dry weight of the plant world is carbohydrate in nature, particularly cellulose, hemicellulose and lignin. Carbohydrates are divided into three groups, namely monosaccharides, disaccharides and polysaccharides. Monosaccharides. Monosaccharides are usually known as simple sugars and possess several hydroxyl groups and a reducing aldehyde or keto group. Monosaccharides having an aldehyde group are called aldoses and those with a ketone group are called ketoses. The monosaccharides are grouped based on the number of carbon atoms present in the molecule. For example, trioses are with 3 carbon atoms. They form the simplest monosaccharides. Example, the aldehyde form glyceraldehydes and ketone form dihydroxyacetone. Tetroses have 4 carbons, for example, erythrose and erythrulose. Pentoses with 5 carbons, example, ribose and ribulose, and hexoses are 6 carbon sugars, example, glucose, fructose and galactose. Disaccharides. Disaccharides consist of 2 sugar units joined by glycosidic bond. In nature, sucrose, glucose, fructose, lactose, galactose, glucose and maltose, glucose are the most common disaccharides. The interacting groups involved in the formation of glycosidic bond are the hydroxyl groups and a molecule of water is eliminated in this process. Sucrose. Sucrose or cane sugar is a major component of the sap of plants. It is made from alpha D glucose and alpha D fructose units. Sucrose C12H22O11 is a disaccharide. Hydrolysis by the enzyme invertase yields invert sugar, so called because of the hydrolysis. Maltose. Maltose or malt sugar is a disaccharide formed from 2 units of glucose joined with an alpha 1 to 4 linkage. It is the second member of an important biochemical series of glucose chains. The addition of another glucose unit yields maltotriose. Further additions will produce dextrins, also called maltodextrins, and eventually starch. Lactose. Lactose, also referred to as milk sugar, is a sugar which is found most notably in milk. Lactose makes up around 2 to 8% of milk by weight. It is the only significant sugar or carbohydrate of animal origin. Lactose is a disaccharide that consists of beta D galactose and beta D glucose fragments bonded through a beta 1,4 glycosidic linkage. An enzyme, lactase, is essential for digestion of lactose, and a majority of adults in this country do not have lactase, which digests milk, sugar or lactose. Polysaccharides. Polysaccharides are long carbohydrate molecules of monosaccharide units joined together by glycosidic bonds. Homopolysaccharides contain one type of monosaccharide, while heteropolysaccharides contain different types of monosaccharide units. Starch present in plants and glycogen present in animals are composed of glucose units. They are important storage molecules for chemical energy. Starch consists of amylose and amylopectin. Dextrin is a storage polysaccharide found in yeast and bacteria. Cell walls of bacteria and plants are often constructed from polysaccharides. Starch. The starches occur widespread as a reserve carbohydrate in tubers such as potatoes, in many fruits, grains and seeds. It has two components, amylose and amylopectin. Most starches contain 80 to 90% of amylopectin and 10 to 20% amylose. Amylose and amylopectin can be separated by taking advantage of the difference in solubility in the water. Both amylose and amylopectin are polymers of glucose, and upon hydrolysis with acid they give D-glucose as the product. Glycogen. Glycogen is the reserve carbohydrate in animals, and it is found in significant amounts in the liver and muscle. It is made up of D-glucose residues. Upon hydrolysis, it yields D-glucose as the product. Glycogen is a highly branched chain polysaccharide, and it resembles amylopectin in structure. Thus, glucose residues are linked together through alpha 1,4 glycosidic linkages, except at the branch points. The branch is linked to the main chain through alpha 1,6 glycosidic linkages. It is very similar to the structure of amylopectin. Cellulose. Cellulose is the most abundant organic compound in nature, and it is the chief constituent of the fibrous parts of the plants. The purest form of cellulose is usually obtained from cotton. It is a polymer made up of glucose residues. Upon hydrolysis, cellulose yields D-glucose as the product. The glucose residues are linked together through beta 1,4 glycosidic linkages. It is found in plants as microfibrils 2 to 20 nanometer diameter and 100 to 40,000 nanometer long. These form the structurally strong framework in the cell walls. Proteins. The term protein is derived from a Greek word that means primary or holding first place. In most living organisms, proteins constitute nearly 50% of the dry weight. They are macromolecular polymers made up of several amino acids. The elementary composition of most proteins is more or less the same, containing 50 to 55% carbon, 6 to 8% hydrogen, 20 to 23% oxygen, 15 to 18% nitrogen and 0.4% sulfur. Several biologically active substances such as enzymes, hormones, antibodies, muscle fibers, respiratory pigments, membrane receptors, transport systems and structures like hair, nail or proteins. Amino acids. Amino acids are bifunctional compounds containing both a carboxylic acid group COOH and a basic amino group NH2 attached to the same carbon atom. They are building blocks of proteins and are linked together by a peptide bond. These are about 300 amino acids in nature, but only 20 of them enter in protein synthesis. Types classification according to polarity of the side chain are: A. Polar amino acids, in which R alkyl group contains polar hydrophilic, so can form hydrogen bond with H2O or water. In those amino acids, R may contain 1 OH group as in serine, threonine and tyrosine, 2 SH group as in cysteine, third amide group as in glutamine and asparagine, 4 NH group or nitrogen act as a base, basic amino acids as lysine, arginine and histidine, 5 COOH group acidic amino acids as aspartic and glutamic B non-polar amino acids. R is alkyl hydrophobic group which cannot enter in hydrogen bond formation. 9 amino acids are non-polar: glycine, alanine, valine, leucine, isoleucine, phenylalanine, tryptophan, proline and methionine. Peptide bond. A peptide bond is a chemical bond formed between two molecules when the carboxylic group of one molecule reacts with the amino group of the other molecule, releasing a molecule of water H2O. This is a dehydration synthesis reaction, also known as a condensation reaction, and usually occurs between amino acids. The resulting CONH bond is called a peptide bond and the resulting molecule is an amide. Two amino acids joined by a peptide bond are called a dipeptide. Tripeptide contains 3 amino acids. Structure of protein. There are 4 levels of protein structure: primary, secondary, tertiary and quaternary. Primary structure. All proteins have well defined orders in which their amino acids are linked to generate the chain like structure. The characteristic sequence of amino acids in a protein is called its primary structure. This is the most fundamental level of protein structure. A protein may contain one or more than one polypeptide chains having similar or different primary structures. If present, these chains are associated with each other with the help of a weak secondary interactions or disulfide bonds. Higher levels of structural organizations in protein architecture depend on its primary structure. Even a small change in the primary structure of a protein has a potential changing in its function substantially. The example of sickle cell hemoglobin illustrates this fact beautifully. The primary structures of normal adult and sickle cell hemoglobins are the same at all except one position. The single substitution of a glutamic acid, that is GLU residue present in the normal hemoglobin with that of a valine VAL amino acid residue alters the property of the protein to the extent that the longevity of the person having sickle cell disease is significantly affected. Secondary structure. Two recurring motifs, secondary structures, have been observed in proteins, the alpha helix and the beta pleated sheet. The alpha helix is a rod like structure and perhaps is the simplest stable form a polypeptide chain can take. The alpha helix content of proteins can be between 0% and 100%. In the case of L amino acids, an alpha helix which turns clockwise is more stable. The helix is stabilized by hydrogen bridges between the NH and the CO groups of the main chain. A alpha helix is the most stable in the case of small amino acids alanine and in the absence of charges and bulky side chains. Proline cannot form a alpha helix and thus interrupts the chain. The beta sheet is also stabilized by hydrogen bridges. This time between NH and CO groups of different beta strands, alignment either in parallel or anti-parallel, the polypeptide chains beta strands are nearly fully extended. The axial distance between neighboring amino acids is 3.5 angstrom units as compared to the 1.5 angstrom units in the alpha helix. The residues can be located above or below the sheet. Lysine and alanine are the amino acids most suited forming a beta sheet. Examples: the two most common types of secondary structures are called the alpha helix and the beta plated sheet. Tertiary structure. Tertiary structure refers to three-dimensional structure of a single protein molecule. The alpha helices and beta sheets are folded into a compact globule. The folding is driven by the non-specific hydrophobic interactions, the burial of hydrophobic residues from water. But the structure is stable only when the parts of a protein domains are locked into place by specific tertiary interactions such as salt bridges, hydrogen bonds and the tight packing of side chains and disulfide bonds. The disulfide bonds are extremely rare in cytosolic proteins, since the cytosol is generally a reducing environment. For example, insulin has two polypeptide chains that are joined by two disulfide bonds. Quaternary structure. Quaternary structure is the three-dimensional structure of a multi-subunit protein and how the subunits fit together. This structure is stabilized by the same non-covalent interactions and disulfide bonds as the tertiary structure. Complexes of 2 or more polypeptides, that is multiple subunits, are called multimers. Specifically, it would be called a dimer if it contains 2 subunits, a trimer if it contains 3 subunits, and a tetramer if it contains 4 subunits. The subunits are frequently related to one another by symmetry operations, such as a two-fold axis in a dimer. Multimers made up of identical subunits are referred to with a prefix of homo, example a homo tetramer, and those made up of different subunits are referred to with a prefix of hetero, for example a heterotetramer, such as the 2 alpha and 2 beta chains of hemoglobin, for example respiratory pigments. Classification of proteins. Since proteins have different amino acid composition, the type and number of each amino acid present in the protein sequence, the order in which amino acids are linked, size, conformation, 3-dimensional folding pattern and function, their classification becomes a rather difficult task. Although all schemes have some overlapping features, depending on their physicochemical properties and functions, proteins can be classified in different ways. Classification based on composition. Composition of proteins is the most common criteria for their classification. Proteins divide of structural constituents other than amino acids are called simple proteins. If a protein is associated with a non-amino acid component, it is named as conjugated protein. These two groups have been further subgrouped as simple proteins, example albumins, globulins and protamines, and conjugated proteins, example glycoproteins, lipoproteins, nucleoproteins, metalloproteins, flavoproteins and phosphoproteins. There are several techniques to identify, separate and quantify the protein like electrophoresis, gel filtration, ion exchange chromatography, high performance liquid chromatography HPLC, affinity chromatography, ultracentrifugation, Lowry-Folin-Seo-Kaltov method, Biuret method and Nin-Hedin reaction. Biological importance of proteins. Proteins are the major structural components of the cell membrane. The membrane associated proteins consist of intrinsic proteins and extrinsic proteins. Many enzymes and enzyme systems are associated with membrane proteins, that is, components of electron transport system. Enzymes are proteins produced within an organism which are capable of catalyzing specific catalytic reactions. They are biocatalysts, which influence the rate of a chemical reaction, usually without undergoing any change themselves. Several hormones are peptides and proteins. They play an important role in regulation of metabolic reactions. The blood proteins include plasma proteins and hemoglobin. Plasma proteins are mainly albumins, globulins and fibrinogen. Albumin maintains the colloid osmotic pressure of plasma and transport materials. Globulins inhibit proteolytic enzymes and help to immune system of the body. Fibrinogen helps in blood clotting. Lipoproteins are concerned with the transport of a fat in the blood. Hemoglobin is a conjugated protein consisting of globin and heme. Hemoglobin carries oxygen in the form of oxyhemoglobin in blood. Some antigens and antibodies are peptides also. In summary, a biomolecule is any molecule that is produced by a living organism, including large macromolecules such as proteins, polysaccharides, lipids and nucleic acids, as well as small molecules like metabolites. In this lecture, we discussed about proteins and carbohydrates. Proteins are important for body construction. These serve a variety of functions within the cells. Some are involved in structural support and movement, others in enzymatic activity. Indeed, the functions of individual proteins are as varied as their unique amino acid sequences. Carbohydrates play an important role in tissue metabolism, providing energy and regulation of blood glucose, sparing the use of proteins for energy. Others are mentioned in part 2